Students, we are discussing chapter mineral nutrition, right? In this mineral nutrition chapter, go for the next topic. There is a symbiotic nitrogen fixation in legume plants. Symbiotic nitrogen fixation in legume plants. Several symbiotic nitrogen fixing associations are known. The most prominent among them is Rhizobium bacteria and legume plant association. Rhizobium bacteria and legume plant association. Okay, right? Several symbiotic nitrogen fixing associations are known. Okay, most prominent among them is Rhizobium bacteria and legume plant association. Rhizobium bacteria and legume plant association. See this statement? Sir, nostoc is associated symbiotically with the thallus of Anthoceras. Nostoc is symbiotically associated with the thallus of Anthoceras. Symbiotic association. Anabina is symbiotically present in the leaves of Azolla. Symbiotic association. Rhizobium bacteria is present in the root nodules of legume plants. Symbiotic association. Like this, several symbiotic nitrogen fixing associations are known, in which the most prominent is Rhizobium bacteria legume plant association. Sir, during this symbiotic association between Rhizobium bacteria with legume plants, root nodules are formed. What are formed, sir? Root nodules are formed. Okay, during symbiotic association of what? Rhizobium bacteria with what? With root nodules. Okay, with roots of which plants, sir? Legume plants. Okay, with what, sir? With roots of legume plants. Okay, what are formed? Nodules are formed. I am writing this word here, nodules. Okay, nodules are formed. See this statement once. Sir, Rhizobium bacteria is symbiotically associated with roots of legume plants. During this symbiotic association, what are formed? Nodules are formed. What are formed, sir? Nodules are formed. Sir, take a pulses plant. Pulses, gram. I am not saying gram in AC. Gram, G-R-A-M. Okay, or take a legume plant. Okay, pulses or gram or legume plant. One and the same, no? You uproot it. Uproot. It means we are plucking the plant from the soil. And if you see the roots when just before flowering, on the taproot system, Fabaceae family, no? Which root system? Taproot system. On taproot system, what you are going to observe, sir? Small, nearly spherical structures are observed. That small, nearly spherical structures are called as what, sir? Nodules. What we are doing here? We are taking a legume plant, nothing but pulses or gram plants. We are uprooting it, plucking the plant from the soil when just before flowering. It means just the plant produces flower, just before the plant produces the flower. Then on the taproot system, what we are observing, sir? Small, spherical, nearly spherical structures called as what? Nodules. These nodules are formed on primary root, secondary root as well as tertiary root. Primary, secondary and what's a tertiary roots. Okay, right? I am writing this statement. Right, on uprooting, on what's a uprooting a pulse plant, even we can call this pulse plant as a gram plant or legume plant, okay, right? Therefore on uprooting a pulse plant, when sir? Just before what? Flowering. Just before flowering. What are observed? On taproot system I am writing. On taproot system, okay, small, what more? Nearly spherical, small, nearly spherical structures are observed. These small, nearly spherical structures are called as what, sir? Nodules. Sir, these nodules are seen on which root system? Taproot system. That is, we can see it on primary root, secondary root, even on tertiary roots. Is this statement okay? Right? Now, listen this statement. Sir, if you see this nodule of legume plants, if you see the central portion of nodules of legume plants, they contains a pigment by name leg hemoglobin. Contains a pigment by name what, sir? Leg hemoglobin, leguminous hemoglobin. Okay, central part of what, sir? Central part of nodules of which plants? Legume plants. Sir, take a legume plant and see the central part of the nodule. Okay, right. Therefore, the central portion or part of nodule of legume plant is red or what in color? Pink in color. Red or pink in color. Right. Why, sir? Due to presence of a pigment. Sir, enzyme or pigment, sir? Pigment. What is the name of that pigment, sir? Leg hemoglobin. Sir, why we are calling it as leg hemoglobin? Because present in legume plants. So, leg hemoglobin is also called as a what, sir? Leguminous hemoglobin. What it is called as, sir? Leguminous hemoglobin. Sir, what this leguminous, that is leg hemoglobin do, sir? It acts as oxygen scavenger. Acts as a what, sir? Oxygen scavenger. Sir, oxygen scavenger means removes oxygen from the surroundings of nitrogenase enzyme and creates anaerobic condition. If oxygen is removed, condition is aerobic or anaerobic? Anaerobic condition. Sir, why anaerobic condition should be created? Because in the root nodules of legume plants, rhizobium bacteria is present. In that rhizobium bacteria, nitrogenase enzyme is present. This nitrogenase enzyme is sensitive to oxygen. It means it cannot fix the nitrogen in presence of oxygen. So what should be removed from the surroundings of this nitrogenase enzyme? Oxygen. Who is removing that oxygen? Leg hemoglobin. So this leg hemoglobin is called as oxygen scavenger. Okay, therefore leg hemoglobin acts as oxygen scavenger. Why, sir? As it removes what, sir? Oxygen from surroundings or vicinity. From where, sir? Surroundings of nitrogenase enzyme. Okay, sir, as it removes oxygen from the surroundings of nitrogenase enzyme, this leg hemoglobin is creating which conditions? Anaerobic conditions. I am saying anaerobic, not aerobic. Therefore, creates which conditions, sir? Anaerobic conditions. Creates anaerobic conditions, right? Right, what is the pigment name? Pigment or enzyme? Pigment. What is the pigment name? Leg hemoglobin, right? Leg hemoglobin. Sir, in this leg hemoglobin pigment, in what, sir? Leg hemoglobin pigment. In this leg hemoglobin pigment, this heme part is synthesized by rhizobium bacteria. This heme part, who is synthesizing this heme part, sir? Rhizobium bacteria. Synthesized by whom, sir? Rhizobium bacteria. Rhizobium bacteria. What left, sir? Globin. Globin is a protein, right? Globin protein. Who is synthesizing this globin protein then? This globin protein synthesized by the host legume plant. Synthesized by whom, sir? Host legume plant. Synthesized by the host legume plant. Okay, right? Therefore, sir, hemoglobin pigment, in this hemoglobin pigment, heme part is synthesized by rhizobium bacteria, whereas this globin protein is synthesized by the host legume plant. Synthesized by the host legume plant. Okay, right? Now listen this statement. Sir, we are discussing symbiotic nitrogen fixation in which plants, sir? Legume plants. Okay. Symbiotic nitrogen fixation requires three components. Symbiotic nitrogen fixation requires how many components, sir? Three components. Requires three components. Okay. What are the three components, sir? Number one, strong reducing agent. Strong what, sir? Reducing agent. Sir, who acts as a strong reducing agent here? Strong reducing agent. Who acts as a strong reducing agent? NADPH. In books, in some books, it will be written as NADPH plus H plus. Okay, or NADPH2. Okay. Sir, from where we are getting this NADPH, sir? This NADPH we are getting from where, sir? Photosynthesis. From where, sir? Photosynthesis. Okay, right? We are discussing strong reducing agent. Sir, even NADH2, right, and FADH2, this can also act as a reducing agent. Sir, we are going to get this NADH2 and FADH2 mainly from where, sir? Respiration. From where, sir? Respiration. Therefore, symbiotic nitrogen fixation requires three components. In that three components, first one is strong reducing agent. Sir, reducing agent, it means it is going to reduce a substance. How, sir? By providing hydrogens. Therefore, strong reducing agent provides hydrogens. Strong reducing agent provides what, sir? Hydrogens. Provides hydrogens. Okay, right? Number two, go for the second component. What it is, sir? ATP. That is ATP energy. What, sir? ATP energy. Right? Now listen this statement carefully. Sir, if organism is free living, take some bacteria which are free living, can fix nitrogen. In such cases, the energy required for nitrogen fixation is provided by the bacteria itself. Sir, take a free living nitrogen fixing bacteria. Free living nitrogen fixing bacteria. Energy required for nitrogen fixation is provided by bacteria itself. Take a symbiotic nitrogen fixing bacteria. In the case of symbiotic nitrogen fixing bacteria, the ATP energy required for nitrogen fixation is provided by the host plant. Provided by the host plant. Is this statement okay? Right? Therefore, we are discussing about symbiotic nitrogen fixation. Yes or no? Therefore, in the case of symbiotic nitrogen fixation, who is providing the energy required, right, for nitrogen fixation, sir? Who is providing this ATP energy, sir? Who is providing this ATP energy? Right, the host plant is providing the ATP energy. Okay, now go for the third component. Number 3, nitrogenase enzyme. Which enzyme, sir? Nitrogenase enzyme. What is the enzyme name, sir? Nitrogenase enzyme. Nitrogenase enzyme. Sir, this nitrogenase enzyme is also called as dinitrogenase. It is also called as what, sir? Dinitrogenase enzyme. Dinitrogenase enzyme. Sir, this nitrogenase enzyme contains 2 units. Having how many units, sir? 2 units. What are they, sir? First unit, second unit. Sir, first unit is made up of molybdenum iron. First unit is made up of what, sir? Molybdenum iron. Second unit is made up of iron sulfur. Second unit is made up of what, sir? Iron sulfur. Sir, this nitrogenase enzyme is required for nitrogen fixation. Nitrogenase enzyme is also called as dinitrogenase. With how many units? 2 units. Unit 1, molybdenum iron. Unit 2, iron sulfur. Okay, therefore these are the 3 components. What? Strong reducing agent which provides hydrogen for reducing a substance. Second one is ATP energy, which is provided by the host itself in the case of symbiotic nitrogen fixation. Third one is a nitrogenase enzyme, which is used for nitrogen fixation, having 2 units. Unit 1, molybdenum iron. Unit 2, iron sulfur. Right? Now, see this diagram once. Sir, in this diagram, right, this is nitrogenase enzyme. Sir, in this nitrogenase enzyme, this curved portion, this is called active site. What is this curved portion called as, sir? Active site. It's called as active site. Okay. Sir, in the active site of nitrogenase enzyme, what came and binded, sir? Nitrogen came and binded in the active site of nitrogenase enzyme. Okay, right? Then what happened, sir? Right, reduction has taken place. What has taken place, sir? Reduction has taken place. Then what is formed here? See here carefully. What is formed here, sir? Right, N2H2 is formed. What is formed, sir? N2H2 is formed. N2H2 is formed. Is it okay? Right? What is formed, sir? N2H2 is formed. Right? Next, you see this diagram once. Sir, this again undergone reduction. When it has undergone reduction, right, when it has undergone what, sir? Reduction. What is formed here, sir? N2H4 is formed. What is formed, sir? N2H4 is formed. N2H4 is formed. Okay, right, N2H4 is formed. Next, this N2H4 has again undergone reduction. Due to this reduction reaction, what has formed, sir? What has formed, sir? 2 ammonia molecules are formed. That is NH3, one molecule, NH3, another molecule. Therefore, 2 ammonia molecules are formed. 2 ammonia molecules are formed. When these 2 ammonia molecules came out as a product, when the 2 ammonia molecules came out as products, right, then the enzyme has become free. It means this enzyme can again participate in the next chemical reaction. Is this statement okay? Right? Therefore, if you see this diagram, right, in this diagram, right, how many protons are utilized, sir? 2 plus 2 plus 2. Total how many, sir? Only 6 protons are utilized. Because how many ammonia molecules are formed, sir? 2 ammonia molecules are formed. 2 ammonia molecules. Yes or no? Okay, right? Now, listen this statement. Sir, mechanism mechanism of biological nitrogen fixation. Mechanism of what's our biological nitrogen fixation in which plants are legume plants. In which plants are legume plants. Okay. Sir, if you take one molecule of nitrogen, I am taking how many molecules of nitrogen, sir? One molecule of nitrogen. This one molecule of nitrogen is reduced to two molecules of ammonia. Two molecules of ammonia. Therefore, biological nitrogen fixation, right? Biological nitrogen fixation, it is a reduction process. Biological nitrogen fixation, it is which process, sir? Reduction process. During this reduction, one molecule of nitrogen is reduced to two molecules of ammonia with the help of 8 electrons, 8 protons, right? And 16 ATP molecules. Okay, 8 electrons, 8 protons and 16 ATP molecules. Sir, this 8 electrons, 8 protons, 16 ATP molecules, they are reducing one molecule of ammonia to two molecules of, they are reducing one molecule of nitrogen, reducing one molecule of nitrogen to two molecules of ammonia. So these 8 electrons, 8 protons and 16 ATP molecules, they are called as which power, sir? Reducing power. They are called as what, sir? Reducing power. As they are reducing one molecule of nitrogen to two molecules of ammonia. Okay, right? Therefore, sir, in this reaction, one molecule of nitrogen is reduced to two molecules of what, sir? Ammonia. Right? What more we got here? H2, right? Plus 16 ADP plus 16 inorganic phosphate. 16 inorganic phosphate. IP means inorganic phosphate. Okay, right? Sir, this H2 can be even written as right, 2H plus. 2H plus. Sir, here we are taken how many protons, sir? 8 protons. But utilized how many, sir? 6 protons are utilized. How many utilized, sir? 6 are utilized. How many came out, sir? 2 protons came out. Yes or no? Therefore, right, out of 8 protons, right, 6 protons utilized. 6 protons utilized. Once you see this reducing power, sir, 8 electrons, 8 protons, what, sir? 16 ATP molecules, 16 ATP molecules. If you take the ratio, how much it will be? 1:1:2. Yes or no? 8:16. 8 ones are 8 ones are 8 twos are 16. Yes or no? Right? Now you take the ratio between what and what, sir? Nitrogen and ammonia. Nitrogen and ammonia. Nitrogen and ammonia. Sir, one nitrogen molecule is producing two molecules of ammonia. One nitrogen molecule is producing two molecules of what, sir? Ammonia. Yes or no? Right? Now see this statement. Sir, one nitrogen molecule is producing two molecules of ammonia by utilizing how many ATP molecules? 16 ATP molecules. Therefore, to produce two molecules of ammonia, 16 ATP molecules are required. It means to produce one molecule of ammonia, how many ATP molecules are required? 8 ATP molecules are required. Yes or no? To produce two ammonia molecules, right, 16 ATP molecules are required, means to produce one ammonia molecule, 8 ATP molecules will be utilized. Is this statement okay? Sir, in this biological nitrogen fixation, energy is utilized. Therefore, biological nitrogen fixation is which process, sir? It is endothermic process or endergonic process. As energy is utilized, it is what, sir? Endothermic or endergonic process. Therefore, biological nitrogen fixation, it is a reduction process. Okay, right? As it utilizes what, sir? ATP energy, right? It is a endothermic process. It's a endothermic process, right? Thank you.